Hello and welcome to Lesson 1, Structure and Function of the Motor, Sensory and Relay Neurons. In this session we're going to look at structure and function of those neurons. We're also going to look at synaptic transmission, and we're going to talk about neurotransmitters and the process of excitatory and inhibitory action. Now we're starting off with the second point on the spec because actually I think it's more important to understand the basic building blocks of your nervous system, of your brain, before we get into the divisions. So I like to go back to square one. Some questions that you might like to think about as we're going through this are: what are neurons? What are the key components of neurons? How is an action potential generated? What is synaptic transmission? What is saltatory conduction? What is excitation and inhibition? And what are the three main types of neurons? Those are some questions to have in your mind as we go through. So this is a neuron. And let's go through and identify the structure. You may have done this at GCSE before. The first structure to notice is the cell body, what we call the soma. Soma means body. This is much like any other eukaryote cell. It contains cytoplasm and mitochondria and so forth. We have the nucleus containing the DNA. We have the dendrites. Now, dendrites are important because they will receive information from other cells that they're connected to. So action potentials will arrive at the dendrites and have an effect on this cell. We have the axon, this long tail-like structure leading away from the cell body. We have the myelin sheath. This is largely made up of Schwann cells, which act as a kind of insulation. And then we have the nodes of Ranvier. Now these are interesting, we'll come back to these in a moment. And then right at the end we have the axon terminals, where the synaptic transmission actually occurs. An action potential is generated if enough signals arrive at the dendrites of this neuron. The action potential then travels across the cell body and along the axon until it arrives at the axon terminals, and that's when the fun begins. So, the membrane of the neuron allows electrical conduction. These are called action potentials. We refer to a neuron as firing if an action potential is generated. An action potential begins at the dendrite, travels across the soma and along the axon towards the axon terminal. Action potentials can occur at different frequencies, and frequency, remember, is the time in between pulses or signals. In this case, the different frequencies characterize different types of cognitive activity, so language, movement, etc. And when we get to EEGs, we'll look at some some behavior that's typically characterized by specific frequencies and amplitudes. This is what is occurring in the action potential, where we see the spike. This is the neuron firing and then returning to its resting state. An action potential is essentially a unit of information. So activity in the visual cortex is characterized by lots of neurons in the visual cortex firing, and that's associated with vision. So every time you see something, neurons in your visual cortex will go like pew, 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 and they're all firing, and they are generating activity because they're actually generating vision. Saltatory conduction then. I said I'd come back to the nodes. One of the structures are these nodes of Ranvier, and these effectively allow the impulse to jump across them as it travels along the axon. Now this has the very interesting effect of actually speeding up the action potential, allowing faster transmission. Now that's important because if it's fundamentally a unit of information, an action potential, then speeding up the transmission of that actually speeds up communication between those cells. And that means that results in faster cognition. Now, why is that important? Think about how complex the human brain is. It's the most complex known structure in the universe. it has to communicate with memory, with your left toe, big toe, back and forth, constantly bombarded by information that it has to filter out and deal with. In order to do that, we need to speed it up. The human brain is much like a really busy international airport. It's Heathrow. You get there, you're rushing, you got your baggage, you need to get to your plane as quick as possible, you can't miss it. Whereas you compare that to something a bit more rural, there's one in Rochester, you take your time, you enjoy the sight. So that's the difference. Now, some animals, some more primitive animals, do not actually have these nodes, and they rely on continuous conduction, which has a disadvantage of being slower. Therefore, you could argue that human information processing is much faster than in many more primitive animals. So, this is when the magic happens. As the action potential arrives at the presynaptic neuron's axon terminal, this is one of the terminuses, it stimulates the release of neurotransmitter into the gap. These diffuse across the gap and bind with receptors on the postsynaptic neuron. So, interestingly, most neurons are not physically connected. There are a few that are, but most neurons rely on chemical transmission to take the signal from the pre to the postsynaptic neuron. They use neurotransmitters to achieve this. Neurotransmitters are chemicals stored in the presynaptic neuron in these vesicles and released through excitosis into the gap, where they diffuse and bind with receptors on the postsynaptic membrane. These receptors have a sort of lock and key shape, very similar to enzymes, if you remember. So the right neurotransmitter will fit the receptor, just so. Neurotransmitters do not bind permanently. It's a short period and then they will detach, and one of two things can effectively happen to them then. They can either be broken down by enzymes present in the gap, or they could be pumped back into the cell from which they came via these channels, these transporter channels. We call this reuptake. When the binding does occur to these receptors, it alters the biochemical nature of the membrane, and if enough neurotransmitter binds, it can generate an electrical potential, an action potential, which is the nerve impulse. Nerve impulses will only trigger, though, if enough neurotransmitter travel across the gap, bind to receptors, and have a significant biochemical effect on the postsynaptic membrane. Let's have a look at a practical example then. So, SSRIs we've covered when we looked at OCD. They are selective serotonin reuptake inhibitors. Their function is to increase the amount of serotonin present at the synaptic cleft, at the synapse. Now, we can't just inject you with more serotonin and increase your levels that way. So what we do is very clever, actually. We prescribe you a drug like Prozac or something like that, which will effectively block the reuptake channels. So the action potential comes along. It stimulates the release of serotonin, let's say. Serotonin diffuses across the gap and binds to receptors. However, it then detaches and gets broken down, or some of it gets pumped back in. However, we've taken SSRIs, and what these are doing is these are blocking the reuptake channel. That effectively means that no more reuptake is occurring. This means that there is more neurotransmitter in the gap and more being dumped into the gap than there would be otherwise, and it's in there for longer. And that will increase the chances that more of these neurotransmitter molecules will actually diffuse across the cleft and bind with the receptor, effectively making more serotonin available here without increasing the overall amount in the system. So we're blocking the reuptake channel, we're essentially closing the exit, saying you can't get back in, entrances, you can't get back in, you're stuck. You're stuck in the gap until you either bind with these receptors or you get broken down some other way. This is a really important example because one of the issues with OCD, as you know, is obsessions, obsessive thoughts, the result of an overactive nervous system, essentially, especially activity in the brain. So your brain is overactive. Serotonin has an inhibitory effect. So by increasing the amount of serotonin in the system, it's having an overall dampening effect, reducing the obsessions, and therefore reducing the anxiety associated with those, and thus reducing the need to fulfill your compulsions, carry out your compulsions to alleviate that anxiety. So it's an ingenious system at work. Why have synapses though? Why not just have directly connected nerve cells? Well, nerve impulses don't always fire, and that's a very good thing. Think about right now, all the sensations you're not currently aware of. Like I'm not currently aware, I am now because I've just thought about it, but I wasn't currently aware of the feeling of the chair on my legs. I'm not aware of the feeling of my clothes on my body. There's a little hum in the background that I was pretty much ignoring, and all sorts of other sensations. None of this is significant enough or necessary for me to take any particular interest in right now. However, if a bear jumps through the window, now I'm interested. Now I've got some purpose to attend to it. So one of the brilliant ways of dealing with this system is by having a sort of threshold over which sensations have to get before they trigger activity in the nervous system or the brain, and the brain. And one of these is this idea that you have to have enough neurotransmitter to diffuse across the gaps, all ganging up on the cell, essentially trying to activate it, and if enough of them do, then it will cause an action potential. Now, an action, a synapse can be defined more or less in one of two ways. It can be said to be inhibitory or excitatory. An excitatory synapse is one whose primary neurotransmitter has excitatory properties, so something like acetylcholine. Whereas an inhibitory synapse is one that predominantly has something like GABA, an inhibitory neurotransmitter. And this means that when that cell is activated, if it releases the excitatory neurotransmitter, it will increase the chance of the postsynaptic neuron firing, or vice versa. Let's have a look at how this works. So, inhibitory, let's say GABA. This is an inhibitory one, it means it will reduce the likelihood of the postsynaptic neuron from firing. So it binds with the PSN, it stabilizes the membrane rather than destabilize it and changing the biochemical, and it reduces the chance that it's going to fire. If there's enough GABA in your system and enough GABA synapses, and they fire, this will have an overall calming effect on the nervous system. Excitatory, such as neurotransmitters such as acetylcholine, these bind to the postsynaptic receptor and increase the chance of a action potential occurring. So inhibitor reduces the chance, excitatory increases the chance. Now, what happens if you've got one inhibitory synapse, maybe this bad boy here, and one excitatory synapse? Well, that's 50-50, isn't it? Will it fire, won't it fire? Well, we can work it out, and we use a process called summation. Summation, which has its roots with the same word as sum, i.e. to add up, literally means that you take the number of excitatory synapses and inhibitory ones, and if they all fire, and they're all bringing in signals, then you can work out whether or not this cell will fire as well. So, excitatory neurotransmitters cause what we call excitatory postsynaptic potential, or EPSPs, and inhibitory ones cause inhibitory postsynaptic potentials, IPSPs. Now, you do need to know this if you want an A. This is detail that you have to know. The summation, if we add up, in other words, excitatory and inhibitory synapses and the effect, we can work it out. In other words, if there's more excitatory, the neuron will most likely fire. Not 100%, but most likely. If the IPSPs are greater than the EPSPs, then it will not fire, most likely. One more thing about it, so, if you've got that in your head and you don't want to shake it, move on. But there are two signals at work here. These are not discrete signals. These are not on and offs. These are analog signals. So think about how much neurotransmitter is passing across this synapse and this synapse and this synapse. Will all have an analog effect. What I mean by that is it will have a sort of amplitude. However, the cell can only fire or not fire, Hamlet style, it either fires or it doesn't fire. And that is it. It can't half fire or fire a little bit. It fires or it doesn't fire. So all these analog signals either generate an action potential or they don't. This cell essentially turns what is an analog signal into a digital one. A digital one is one that has discrete states, i.e. on or off. An analog one is one that can have many different states, such as different volumes and different speeds or whatever. That is summation, and it's really clever. Just one more point about this before we move on. Imagine that you've got different connections to this particular neuron, and some are really being stimulated a lot. So again, a lot of action potentials at high frequency from this synapse here, and we've got inhibitory ones here, but far fewer, less frequent. Well, this is going to have a stronger effect on this cell than this one. So that's what I mean by analog to digital. And talk to me about my ordering pizza example later. Different types of neurons there. So just to recap, we've talked about the neuron, we've talked about different parts of it, and we've talked about how neurons communicate with other neurons. Now we're going to look at different types of neurons. Your nervous system is made up of 3 different types: sensory, relay, and motor. Now, sensory neurons carry nerve impulses from receptors in the peripheral nervous system, i.e. in the skin, in the eyes, wherever it may be, to the central nervous system, right? So they're carrying signals from the outside in. However, relay neurons present in the central nervous system take that information and do something with it. They either relay it to the brain or they relay it to the motor neurons. The motor neurons carry information from the central nervous system out to the muscles. So sensory neurons detect the threat, relay neurons will relay the message, and motor neurons will deal with the threat, essentially. Now, there are 3 very different types. So motor neuron is very similar to the one that we looked at the beginning. It has this long axon and the cell body at one end. Sensory neurons tend to be a little different. So they'll have a cell body in the middle, essentially, and 2 long, a long axon that spans across it. And then relay neurons tend to be shorter and dumpier, at least they look that way in diagrams, with quite a large soma and lots of dendrites, but not a particularly long axon. Here's another way that they're sometimes depicted, because if you're looking in textbooks it can be confusing. So this would be a motor neuron, this one's a sensory neuron, and this is what we call this relay neuron and interneuron. What's the difference between a sensory neuron and the interneuron? Size, it seems, in this diagram only. So those are the 3 types: sensory from the outside in, relay to the brain or to the muscle, to the motor, and then motor to the muscles to do something about it. I'll show you some diagrams. Sensory neurons then, they transmit signals to the central nervous system via the spinal cord. Relay neurons in the central nervous system, spinal cord, connect sensory neurons to the motor neurons. So they go between guys. I'll show you a little diagram of this in a minute. Motor neurons then carry the signal to the muscles from the central nervous system. So let's have a look at this. This is a reflex arc. If the doctor's ever hit you just below the knee, you've got a reflex there, which you don't have any control over, which can be activated. So sensory neurons detect the strike, and they send the information to, this is a slice through, top down, of the spinal cord. So the sensory information is sent through to the spinal cord, and here you can see it's connected to relay neurons. These relay neurons themselves are connected to motor neurons, or what we call the efferent division of it, and they send a signal to the muscles to do something. In this case, this sends a message to this muscle to contract and pull the leg up, and this one to relax. Let's have a look at another example. I'll just move me down here. Many of these reflex arcs do not require communication with the brain. They don't require instruction, it's instant. So the hot pan. Receptors in your hand send a signal that the pan is hot to the central nervous system. A decision is instantly made. Instantly is a bit far, and this via relay neurons connects to your motor neurons to pull your hand away. However, the relay neurons also send a signal to your brain that says, ouch, this is painful. So this signal is actually sending pain, even though it's not requesting information from the brain, it's not requesting instructions, it's not saying, should I move my hand or should I leave it? It's already going to do that as part of the reflex. It is sending this signal saying, ouch, this is painful, isn't it? Right, so that is how our nervous system works. We've covered neurons, we've covered synaptic transmission, and we've looked at different types of neurons present in the body. We will come across all of this in the next lecture again, but here are some exam questions to get you in the mood. We have a really easy question that you could get right at the beginning of the exam paper to build your confidence. Which one of the following is found in the central nervous system, the motor neuron, the sensory neuron, or the relay neuron, or the synapse? And explain the nature of synaptic transmission and evaluate changes in neurochemistry of the brain as an explanation of behavior. Now, the first one's quite easy. Hopefully you know the answer. I'm not going to tell you what it is. You can tell me next lesson. This question, though, is a bit trickier. The AO1 for this question is present in this lecture. You could just write and describe, in my words, essentially, how this has occurred. Use your own words. You need to understand this. But if you did, that would be fine. However, it's a 12 marker. So where are you getting your evaluation from? Your evaluation is actually AO2. Because what you will need to do is apply this to some sort of behavior that you know about. And the behavior I would recommend you apply to for now, that you might talk about schizophrenia later, is OCD and the effect of SSRIs, which is why I included that in this lecture. So your AO1 would be how synaptic transmission occurs and blah, blah, blah. And then the O2 would be SSRIs and how synaptic transmission, low levels of serotonin, high levels of dopamine can have behavioral effects. OCD is your go-to girl for that one. So, I hope you have got something out of this lecture. We've looked at sensory receptors in peripheral nervous system, relaying signals to the central nervous system, done through action potentials. Different neurons are responsible for sensing and acting. Neurons are connected via synapses. So now you know what neurons are, you know what the key components are, how action potentials are generated, synaptic transmission, substrate conduction, excitation and inhibition, and what are the three main types of neurons. Make sure you've annotated your notes before you come to lesson, and we'll do lots of application next time. As always, thank you very much, and I will see you around. Bye.